The further and further we get into the molecular genetics side of this course, the more excited I get. This is really where my passion lies, is what the DNA is doing, how the DNA is being manipulated and controlled by the cell, and the impact that has on the behavior of the cell, which really, when we say behavior of the cell, we mean cellular phenotypes. So we're going to spend this lecture talking entirely about how bacterial cells control their genes, control the expression of their genes, control the proteins that they make from those genes. And in this course, we're going to completely ignore eukaryotic gene regulation. Eukaryotic gene regulation is covered very much in depth in Bio 320, which is cell and molecular biology, but we will not cover eukaryotic regulation here. It's far too complicated. This is chapter 16 from your textbook, and today we're going to just begin to introduce the basics of gene regulation so that we're all clear what we mean by that. We'll introduce the idea of operons in bacteria. This is how bacteria organize and arrange their genes. We'll define and describe negative and positive control of genes. Then we'll use two examples of this in order to put it into a real living context, the lac operon from bacteria as well as the trip operon from bacteria. And we'll focus on the phenomenon called attenuation, which is how the trip operon is regulated. Hopefully by now, after watching the last lecture, everybody is comfortable with the whole point of everything. The idea that genes do govern and control traits, but they do so indirectly by encoding proteins. What happens is that the DNA genes are transcribed into messenger RNAs. The messenger RNAs are then translated into proteins. The proteins float away from the cellular machinery and do whatever it is that that cellular machine, that protein, was meant to do. The proteins are the doers, the machines of the cell. So once the protein is made, it is free to do that thing. Whatever that thing that the protein should be doing is, that activity gives the cell a new individual trait. I've just again described the central dogma. DNA is transcribed into RNA, and RNA is translated into protein. DNA is the information warehouse for how to build proteins. Proteins are the actual machines that keep the cell running and alive. But we need to keep in mind that the making of RNA, the act of transcription, is very, very expensive in the currency of cellular energy, in ATP molecules. Cells are striving to do everything they can to spend as little ATP as possible. ATP is precious. Energy is precious. And so transcription is expensive and should only be done when it's needed. The same can be said of making proteins. Making proteins is also extremely energetically expensive. On top of that, some proteins are only good to have around. Some machines are only needed to do their thing when they're actually needed. And the flip side of that coin then is also true. Sometimes, when you don't need a particular function in the cell and you don't need the protein around to do that, having that protein around could be a bad thing. So all of these things together mean that we require a way to regulate gene expression. We require a way by which we can turn genes off so that proteins cannot be made from them, but then turn genes on when we do need those proteins. If we could regulate genes, we could make sure that we only make the proteins that we need when we need them, and we don't make those proteins when they're not needed, and more importantly, save all of that ATP and all of that energy by not transcribing genes we don't need and not making proteins we don't need. So we avoid the conflict of having a protein around that is going to harm us when it's around the wrong place at the wrong time, as well as saving ATP because we're not making proteins we don't need. So I'd like you to take a brief moment and just imagine this scenario. This is true. E. coli can metabolize almost any sugar on the planet. By metabolize, we mean extract energy from. Eat. So E. coli can eat almost any sugar that's available, and E. coli can make all 20 amino acids that it requires for its own proteins. But metabolizing sugars other than glucose requires energy. It's actually expensive to eat sugars other than glucose. This is true of E. coli, this is true of ourselves. All living things on this planet prefer glucose as an energy source above all others. Glucose is the cheapest, easiest to use energy source. And making amino acids is expensive for a cell to do. making amino acids takes time and energy. So if you have amino acids on hand already, there's no need to make more. So from E. coli's perspective, E. coli only wants to metabolize these other difficult-to-use sugars when those sugars are around and when glucose is not. Think about that. Allow that to make sense for you. E. coli would love to have glucose. Glucose is its favorite food. If there's no glucose around, E. coli could starve and die. If there's no glucose around but there's another sugar around like lactose or fructose or galactose, E. coli would rather eat those more complicated, difficult to digest sugars than die. So when those other sugars are around and glucose is not, E. coli would prefer to metabolize those other sugars. However, if those other sugars are around and glucose is around too, E. coli would prefer to consume the easy-to-digest glucose and ignore those other sugars. Also from E. coli's perspective, E. coli can and wants to make all of its amino acids when those amino acids are not available in the environment. However, if amino acids are available for E. coli to scavenge and reuse, it wants to avoid going through the energetic hassle and trouble of making them from scratch. The metabolizing of these sugars requires proteins that are encoded by genes. The metabolism and synthesis of these amino acids requires proteins that are encoded by genes. So when we say things like E. coli wants to avoid some metabolism while preferring another, when we say things like E. coli wants to avoid making amino acids when amino acids are available in its environment, we are talking about reversible regulation of genes. We're talking about turning genes off when the proteins they encode are not needed. So when those amino acids are around in the environment, we want to turn off the genes that encode the proteins for making amino acids from scratch. But when those amino acids from the environment become depleted, and E. coli now needs to make its own amino acids, we would like to turn those genes on to make those proteins so that new amino acids can be synthesized. When those amino acids have built up and their levels are high again, we'd like to turn those genes off again because we don't need those protein machines anymore. And when amino acid levels become depleted, we'd like to turn those genes on again. What we're really getting at here is that we would like to, as living cells, have our genes on light switches where we can toggle them on when we need them on, off when we need them off, on again when we need them on, off again when we need them off. Reversible regulation. We want proteins on demand, so we require transcription and translation to be regulated. Today what we're doing is discussing this phenomenon, discussing this type of regulation in bacteria. Most of the general concepts and principles we talk about today apply to eukaryotes. What's different between prokaryotes and eukaryotes is the how the regulation is achieved. But the concepts, the logic is the same. The molecular definition of a gene, and this is going to be the definition that we now adopt, is simply any DNA sequence that is transcribed into RNA. Any DNA sequence that is transcribed, or expressed, as we would say, is a gene. We have structural genes, and structural genes are defined as those that encode structural proteins. Well, what are structural proteins? Almost every protein in the cell is a structural protein. Proteins that are needed for metabolism, such as breaking down sugars, are structural proteins. Proteins that are needed for biosynthesis, such as making amino acids from scratch, those are structural genes. The proteins that are truly structural, the proteins that make up the microtubule cables that we talked about in mitosis, that make up the rest of the cellular scaffolding, those are also structural. All of these are structural proteins encoded by structural genes. The other category of genes that we'll discuss today are regulatory genes. Regulatory genes are those genes that encode regulatory proteins. Regulatory proteins, and there are such things as regulatory RNA molecules as well, that are made by transcription. But these regulatory proteins are proteins that somehow interact with the DNA to affect and regulate gene expression. So there are proteins encoded by genes that turn other genes on, and there are proteins that turn other genes off. Those proteins are regulatory proteins, and they themselves are encoded by regulatory genes. Many times these regulatory genes encode regulatory proteins that directly bind to DNA, and by directly binding to DNA, they either promote gene expression, turning genes on, or inhibit gene expression, turning genes off. Some genes encode proteins that are unregulated, because these are proteins that are needed by the cell all of the time. There are proteins that cells always require, and we call these genes constitutive. Unregulated genes that are always on are constitutive genes. Now there's plenty of DNA in any living cell, even in E. coli, that's not transcribed. If the definition of a gene that we are now using is a transcribed DNA region, what is this DNA that is untranscribed? Well, lots of the untranscribed genomic DNA, especially in bacteria, is made up of regulatory elements, different than regulatory genes. So regulatory elements are DNA sequences that are not transcribed, but instead somehow affect and regulate the expression to the genes, the expression of the genes to which they are linked. It's getting pretty confusing, I think. What we're saying here is when there is a regulatory protein and it binds to DNA, let's say it's to promote the expression of a gene, we are seeing all of these players come together. What we're seeing here is a bacterial gene. This portion right here is the protein coding region of this gene. In here, on this DNA, is the instructions for how to build a protein that can manifest if we transcribe this gene into a messenger RNA and then translate that messenger RNA into a protein with a three-dimensional shape. This is the gene. Lying here, just in front of the gene, is a regulatory element. It's a DNA sequence, just like any other. But this DNA sequence doesn't contain the information for building proteins. This DNA sequence is a landing site, or a binding site, for another protein. This is the regulatory element. It's sometimes referred to as an E-box because it is a regulatory element, E for element. The protein that we see here, binding directly to this regulatory element, is a regulatory protein. This protein, when it is made by the cell and binds to this element, promotes or drives the expression of this gene. This regulatory protein is encoded by its own DNA gene, which we would refer to as a regulatory gene. This gene is somewhere else on the bacterial genome. The regulatory gene is expressed, the regulatory protein is made. The regulatory protein binds to the regulatory element. When this binding occurs, we get the activation of this gene, transcribing it to a messenger RNA and translating it into a protein. If the regulatory gene is not expressed, we will not make the regulatory protein. If the regulatory protein is not made to bind to the element, we will not transcribe this gene. Without transcription, we cannot make the protein, and we have thus regulated or inhibited the expression of this gene. If we want to have that reversible regulation and we want to turn this gene on again, we simply make the regulatory protein. Once made, the regulatory protein binds to the element. This gene is turned on, transcribed and translated, and we make the protein we want from this gene. That's the idea of reversible regulation. So regulatory proteins usually bind to a regulatory element, and there are actually two primary types of gene regulation that occur at the regulation level of transcription, and we coin them positive regulation and negative regulation, or positive control and negative control. Positive control is used or described as when the natural state of the gene is to be off. You can think of it as a default state. The default state of the gene is to be off. and something has to be done to turn that gene on. The cell has to engage in some action to turn that gene on. We refer to this as activation. The gene becomes activated. Negative control is the opposite scenario. In negative control, the default state of the gene, its natural tendency, is to express itself. And the cell has to intervene or take action in order to turn that gene off. And we call that action repression. The word repression is hiding behind this little integrity thing here. So when we repress a gene, we turn it off, and when we activate a gene, we turn it on. We're familiar with these kinds of relationships. We don't have to think about this in the abstract. In my mind, a car is a perfect example of positive control, and a hot water heater is a perfect example of a negative control system. Think of a car. What's the natural default state of a car? If you have a car, or you can look out a window right now and see some cars, look at their natural default state when no one's interfering with them. The natural default state of a car is off. Cars don't really do anything on their own. If you want the car to go, if you want to activate the car, if you want the car to do something, you have to sit down and turn the car on. You have to activate the car. That's positive control. And then the car will do something, but its default state is to be off. Now I want you to think about the hot water heater that is giving you hot water in your dorm room, in your apartment, or in your home. Think of that hot water heater. What's the default state of that hot water heater? Do you need to go downstairs into your basement every time you want hot water and turn that hot water heater on? Or is it more like by some miracle, whenever you turn on the hot water faucet, hot water comes out? The default state of your hot water heater is on. It automatically heats water when needed. And I guarantee you that somewhere in your basement or wherever your hot water heater is located, there's going to be a red labeled power switch. That's an emergency cutoff for the hot water heater. You can flip that switch off, and you can turn the hot water heater off if you need to. If there's an emergency or a leak or it's malfunctioning, you can turn the hot water heater off with that switch. But you have to go and take action to do that. You have to interfere with the hot water heater in order to turn it off. The natural tendency of that hot water heater is to be on, and that's an example of negative control. Genes come in both types. Some genes are like cars. They're naturally off, and they have to be turned on by other proteins. And some genes are like hot water heaters. They're naturally on all the time, and they have to be actively turned off by other proteins. Now, we're focused today on this transcriptional level of regulation, where by controlling whether or not transcription can occur, we control whether or not a gene is expressed. But I'd like to just take one slide here and have you kind of keep in the back of your mind that protein expression can be regulated at every single step in the pathway to making protein. So we can regulate protein expression by simply controlling whether or not the cell can access the DNA at all. If the DNA is very, very tightly wrapped and inaccessible, we cannot express protein from it. If we relax that DNA and make the DNA more accessible, then we have an easier time expressing genes. The process of transcription is a given. That's what we're talking about today. But by allowing transcription to occur, we can allow a protein to be made, and by inhibiting transcription, we block that protein from being made. However, once transcription has occurred, cells can manipulate the stability of the messenger RNA. If they make the messenger RNA very unstable, so that it breaks down very, very quickly and rapidly, you make very, very little protein from that unstable, short-lived messenger RNA. But if you can somehow stabilize the messenger RNA and make it last longer, more and more rounds of translation can occur from it, and you make more and more protein. The actual process of translation can be regulated. Translation can be blocked. Those ribosomes can be blocked. And if translation is not allowed to occur, it doesn't matter how much messenger RNA you have around. Without translation, you will not be building protein. However, if translation is allowed, protein can be made. And then, believe it or not, even after the protein is actually synthesized, it can be regulated. It can be modulated between an active form and an inactive form by modifying the protein itself after it is built. Believe it or not, we've covered 3 different core courses of a biology curriculum. Right now in genetics, we're going to talk about the regulation of transcription. We'll talk about other things like messenger RNA stability and translation regulation when we do Bio 320, cell and molecular biology. And finally, the idea of regulating proteins after they're made by modifying the proteins directly, those are biochemical phenomena that you will learn about when you take biochemistry. So I just want you to appreciate that regulation is not a one-shot deal. The cell is constantly engaged in these checks and balances, constantly monitoring the proteins that it needs and the expense at which it's going to cost to make those proteins. And so the cell is constantly balancing its energy requirements and its response time. Do I need this protein right now? If the answer is yes, I should make it. If the answer is no, I shouldn't. Do I not need this protein right now, but when I do need it, I'm going to need it in a hurry? If the answer to that is yes, then maybe we make the protein now, even though it's expensive, but we modify that protein somehow so that it is not active. And then when we need it, we can just turn that protein on and we don't have to concern ourselves with the lag time of synthesizing it. The cell is constantly asking itself questions like, how expensive is this protein to make? How quickly will I need this protein when I do need it? And adjusting its activities based on the answers of those questions. It's an amazingly complicated process, but amazingly elegant. The cell is doing some ridiculously complicated things at all times, constantly readjusting itself and self-diagnosing itself for its needs in that moment, and also anticipating its cellular needs for the future. But let's get back to transcription now. Let's focus for the rest of this lecture on regulation at that one level of transcription. So if we're talking about prokaryotic cells, bacterial cells, we want to start introducing this idea of operons. We said it before in this course, but bacteria are really awesome little buggers. They're incredibly efficient life forms. Most of their DNA is protein coding. They have a very efficient, streamlined genome. But in addition to that, bacteria have also clustered or organized their genes, the genes of similar function, together into these functional units called operons. Now, operons are a collection of multiple genes that all have a similar function for a similar cellular need, but they're controlled by a single regulatory element. We'll use as our example the lac operon. The lac operon consists of 3 different genes, all of which encode proteins needed to metabolize and feed on the sugar lactose, the milk sugar lactose. These clustered genes are all under the control of a single regulatory element, a single DNA sequence that controls them. We refer to this as a promoter. A promoter is a DNA regulatory region. It's a regulatory element. It's an on-off switch for a gene. The promoter is where the transcriptional machinery assembles. That's the RNA polymerase, the enzyme that makes RNA during transcription, binds to the DNA at the promoter, assembles at the promoter. So if the promoter is active, if it's on, if it's accessible, that means this RNA polymerase enzyme can bind to that promoter. If RNA polymerase binds, we get transcription, period. However, if the promoter is kept inactive, in an off state, in other words, it's not accessible, RNA polymerase cannot bind. If RNA polymerase cannot bind, we cannot get transcription, because RNA polymerase is what makes transcription happen. And without transcription, we cannot express the protein. We can't express the gene. So when these promoters are on, all of the genes of a single operon are transcribed together as a single messenger RNA molecule. But then each individual protein for that gene is translated separately. This schematic, I think, shows that best. The green region here is meant to indicate the promoter. The DNA polymerase enzyme, the RNA polymerase enzyme would bind right here to this promoter. And it would then move down the gene, move down the operon, making a single messenger RNA that is an RNA copy of these 3 DNA genes. In this case, 4 DNA genes. Then at the terminator, the RNA polymerase would fall off, transcription would cease, and the messenger RNA would be completed. This is a single messenger RNA that contains the instructions for building 4 different proteins. Then, once translation occurs with the ribosome, each one of these proteins is going to be synthesized or translated independently. And so we make protein A, protein B, protein C, and protein D separately. I should say that this Operon strategy, both of grouping genes of similar function together in a single location, as well as having multiple genes on a single RNA, this never happens in eukaryotes. This is exclusively bacterial. Now, most of these operons also contain a regulatory gene. Now, remember, regulatory genes encode regulatory proteins, and regulatory proteins bind to DNA and influence gene expression. So, operons typically have a regulatory gene that encodes a protein that's used to regulate the operon, keep it on or keep it off. The regulatory gene is usually not clustered with the others. It is separate, but nearby. It's usually constitutive, so it's expressed all the time, so the regulatory protein is made all the time. The regulatory protein, once made, can, under the right conditions, bind to the regulatory element and either turn the gene on or turn the gene off, depending on how that particular gene is regulated. What is always the case is that the regulatory gene expresses the regulatory protein, and the regulatory protein binds to the regulatory element and somehow influences the expression of the operon. So now we come to a new aspect of positive and negative control. We have to now consider positive and negative control in the context of regulatory proteins. So we've already discussed positive and negative control. Just to remind you, positive genes are in a default state off and need to be turned on. The car was our example. Negatively controlled genes are usually on and need to be turned off, like hot water heaters. But we also have inducible operons and repressible operons, and these terms describe what the regulatory proteins are doing. Inducible operons are kept off by the cell most of the time. So the cell works on the assumption that the operon is not usually needed, and only when that operon is needed does the cell allow it to be expressed. Repressible operons are the opposite. For repressible operons, the cell usually needs the operon on, and so it keeps that operon on most of the time. But sometimes, in rare exceptions, the operon is negatively regulated and turned off by the cell. As much as it may seem counterintuitive, we can have all 4 possible combinations of negative and positive control and inducible and repressible operons. In other words, we can have positive inducible, positive repressible, negative inducible, negative repressible. We'll talk about negative inducible first. Negative inducible operons, meaning an operon that is defaulted on and needs to be turned off actively, yet is usually kept off by the cell and is turned on by the cell when needed. How does that work? Well, for negative inducible operons, the regulatory protein is a repressor, and so the gene's natural unregulated tendency is to be on. Any gene whose default tendency is to be on is being regulated in a negative fashion. The regulatory protein is a repressor, so it turns the genes off. But we're talking about an inducible operon. Inducible operons are usually kept off and only turned on by the cell. So if we have an inducible negative operon, the regulatory protein, the repressor, is always made by the cell. And so the gene is continually repressed. However, when the cell decides that it needs that operon on, it somehow inactivates the repressor, allowing the gene to default back to its naturally on state. I'm always one to see things in pictures better than in words. We're talking about negative inducible. When we say negative regulated, we know that the gene naturally wants to be on. Its unadulterated, unmanipulated state is to be on. But when we say inducible, we mean that the cell keeps the gene off and only turns it on when needed. So if the cell is keeping the gene off most of the time, but the gene's natural tendency is to be on all of the time, the cell must repress the gene with a repressor to keep it off. Here, the repressor is this orange protein here. So normally, under all cellular operating conditions, the repressor is made, it thwarts the gene's natural tendency, and the gene is off. But in the rare exception that we get some signal that the operon is needed, the repressor is allowed to leave the gene, and the gene defaults to its on state, and we get an induction, an inducible expression of this gene by allowing it to default to its natural state. Negative repressible. We're still negative regulation, so the default state of the gene is still to be on. But since we're saying it's repressible, we're saying that the cell normally wants the operon on and only turns it off in the rare circumstance that it wants to turn that operon off. So it's a much easier scenario for negative repressible. The tendency of the gene is to be on. The want of the cell is to normally keep it on. And so normally the cell just allows the operon to be on so that the gene is continually expressed. However, in that rare exception where a cellular signal is received and the cell decides that it doesn't need that operon on right now, only then does the cell intervene and activate a repressor to keep the gene off. So the schematic for this would be the natural tendency of the gene is to be on, and the most common desire of the cell is to leave the operon on, so normally the gene is left unmolested, and so it is on. However, it's a repressible gene, meaning there are times, infrequently, but there are times that the cell wants this operon off, and so on those times it activates or makes a repressor, the repressor binds to the operon, and the operon is turned off. Negatively repressible operons are usually involved in biosynthesis. The default state is on. Normally the cell wants biomolecules to be made, but in the rare circumstance that some biomolecules are available in the environment, it's time to turn those operons off and save some energy. From the cell's perspective, I know I always need adenine. I know I'll almost always want to make adenine. So my genes are naturally on, and I normally leave them on. I've got to keep making adenine. I can't ever stop making adenine. But in the rare circumstance that there's adenine in my environment, and there's plenty of adenine around, I can stop making adenine. And so at that point, I can repress my operon and save some energy. Incidentally, this is also called feedback, where the product of a gene pathway itself stops its own synthesis. So here, the presence of adenine is what turns off the pathway for making adenine. When you have adenine, you don't need to make more adenine, so adenine shuts off its own synthesis. As adenine levels drop, the cell needs to make more, so the absence of adenine is the trigger to make more of it. That'll become much more important as you go on to biochemistry. And so in this case, the schematic that we're showing here, this blue triangle, the blue diamond, would be adenine. Only when adenine is present in the cell does it bind to a repressor protein and allow that repressor protein to bind DNA, turning off the operon. As adenine levels drop, the repressor no longer binds, and so the operon goes back to its default state, which is on. The other two examples, positive inducible. now the natural tendency of the gene is to be off. And inducible means that the cell has to actually go and turn that operon on when it's needed. So here the regulatory protein is an activator. The gene's natural unregulated state is to be off. It has to be turned on. And the cell usually keeps the gene off. The regulatory protein is usually not made. And it's only made or activated when the cellular signal is received that tells the cell that that operon is now needed. So the gene, the operon, is naturally off, usually left off, and only turned on by the cell when needed. So we see here that the gene is naturally off. In the absence of any regulatory proteins, it remains off, but the cellular conditions become such that the operon is needed, and so now the cell has to go ahead and induce that gene by making or activating a transcriptional activator that is going to allow that gene to be transcribed. Finally, positive repressible. Still we're in positive regulation, so still the default state of the gene is to be off. But repressible means that the cell normally has this operon on and has to make conscious steps to turn the operon off. So if the gene's natural tendency is to be off, but the cell usually keeps it on, that means we still have a regulatory protein that is an activator, but the activator is almost always made and active. So the gene is usually continually expressed regardless of its default state because the activator is always around. But when the cellular signal is received that tells the cell that the operon is no longer needed, the activator is no longer made, and the operon is allowed to revert to its default state, which is off. So here we have a positively regulated gene. Its default tendency is to be off, but see, under all normal circumstances, the activator is on and binding to the operon, forcing that operon to be on almost all the time, pushing it against its default state. However, when a cellular signal is received that the operon is no longer needed and transcription can cease, the activator is brought off of the DNA, and the unmolested DNA is allowed to revert to its default state, which is off. That's what positive regulation means. And so we are repressible. The cell turns this transcription off by removing the activator from the operon. I know it's confusing. I can appreciate that. But please review these a number of times. Get comfortable with the differences between positive and negative regulation, inducible and repressible regulation. Really try to own these four concepts, as they will help you not only master those concepts themselves, but also reason your way through the entire regulation of the lac operon. And that's what we're going to spend most of our time on in today's lecture, is using the lac operon as an example for this kind of multifaceted regulation. So we're going to use the lac operon as not only an example to see how these types of regulatory strategies work, but to also get a sense of the physiological context of regulation in this way. In other words, how and why would a cell regulate genes in this way, and what impact does it have on the cell as it tries to survive? All living things on this planet require sugars to survive. Carbon is the mode of energy transfer on this planet, and sugar molecules are nothing more than strings of carbon and water. So all things live on sugar. And the simplest sugar to digest and extract energy from is glucose. So all living cells on this planet, from the simplest bacterial cell to our own cells in our own body, prefer glucose over any other fuel or food molecule. It's the easiest and energetically cheapest sugar to extract energy from. So if you put a plate of glucose in front of a bacterial cell and a plate of lactose in front of a bacterial cell, it can survive on either one. It can metabolize on either of those sugars for energy. But 10 times out of 10, those bacterial cells will choose the glucose plate because it is easier and energetically cheaper to digest. If you don't give bacteria any sugar, including glucose or lactose, it's going to die. It'll starve. If you offer bacteria lactose only, it's no dummy. With a lactose-only diet, the bacteria has a very easy choice to make. Metabolize lactose as a sugar source or die. It chooses lactose. It's a survivor like everything else on this planet is. But if you give bacteria a choice between glucose and lactose, it will choose glucose. And I want to stress that. If you offer bacteria a choice, think of that, bacteria. If you offer bacteria a choice, it makes that choice. And it makes that choice based on high-level concepts such as energetic efficiency and ease of digestion. It's an easy, common-sense concept to grasp. It makes sense from our perspective. If you're starving to death, and I give you a bowl full of honey, or a coconut that you've got to bash on a rock for the next 3 hours to crack open, you're going to choose the honey because it's easier to use and it will satisfy you quicker. The coconut, although it is food, is harder to get to and so more difficult to use as an energy source. That makes sense. What is amazing to me is that the bacteria is making this choice and is making it correctly. So molecularly, if that is a word, which it isn't, how do bacteria make this choice? The way that they make the choice on the molecular level is quite easy. If lactose and glucose are around, bacteria keep their lac operon off. By not expressing the lac operon genes, bacteria make it impossible for themselves to metabolize lactose, because the proteins made from the lac operon are those proteins needed to metabolize lactose. So by not expressing the lac operon and not making those proteins, bacteria render themselves unable to digest lactose. Okay, so that's how the choice is made. Keep the lac operon off, you don't digest lactose, and all you're left with is glucose to digest. The question for us becomes, how do bacteria do that? How do bacteria know to keep their lac operon off when glucose is around? And how do bacteria know that they need to turn their lac operon on as glucose levels become depleted? Well, in order to understand that, we have to first understand the lac operon. There are 3 genes that make up the lac operon proper. That is, 3 genes that are clustered together under the control of a single promoter. They're called LAC-Z, LAC-Y, and LAC-A. LAC-Z encodes an enzyme that is called beta-galactosidase, a protein machine whose job it is to cleave lactose sugar into its 2 basic sugar building blocks, galactose and glucose. So lactose is a disaccharide, if you're comfortable with that term. Lactose is a single sugar molecule made up of 2 simpler sugars, and the 2 simple sugars that come together to make lactose are galactose and glucose. So LAC-Z encodes the protein needed to do that cleaving. As a side note, beta-galactosidase also converts lactose into a molecule called allolactose. That's just an intermediate on the way to being broken down into galactose and lactose. LAC-Y encodes a protein type that we call a permease, as in make permeable or permeate. LAC-Y is a protein, or LAC-Y, the gene, expresses a protein that goes to the bacterial cell membrane and transports and allows lactose into the cell. And LAC-A encodes an enzyme that has a known function, but believe it or not, as much as this operon has been studied, to this day no one really understands how or why this enzyme is needed for lactose metabolism. So we'll focus on LAC-Z and LAC-Y. LAC-Y is needed to get the lactose into the cell, and LAC-Z is needed to break it down into simple sugars. The regulatory protein for this operon, like all regulatory proteins do, it lies a little bit apart from the cluster genes. It's called LAC-I. The LAC-I protein is a repressor, so it can turn the lac operon off when binding to the promoter region. And the lac operon is an example of a negative inducible gene. It is inducible in the sense that its natural cell-maintained state is to be off, and the cell decides to turn the Operon on when it's needed. And the negative side of that is that the natural tendency of the Operon is to be on, like a water heater, and it is turned off by the cell under most conditions using the Lac-I repressor regulatory protein. So the natural tendency of this Operon is to be on, but it is constantly and continually kept off by the cell through the action of the Lac-I repressor protein. Keep in mind, with cellular systems, there's never 100% efficiency, so although the repression is robust, it is not absolute. And there is very, very low level, like a trickle, drop-by-drop, leaky faucet of expression from the Operon. So at any one time, there are minuscule levels of Lac-Z, Lac-Y, and Lac-A protein expression, but at very, 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 very low levels, because repression is robust. So we're going to first discuss how lactose influences the regulation of the Operon. Let's see how this all works. When there's no lactose around in the cell, we can agree on one thing. When there's no lactose around, there's no need for the Operon to be on. Never forget the point of the Operon. The Operon contains genes that encode proteins needed for the metabolism of lactose. So if the cell doesn't have lactose in it, there's no need for lactose metabolism. There's no need to express the Operon. So when there's no lactose around, the Lac-I protein repressor is binding to a certain region of the promoter, which we call an operator. It's binding to the promoter, blocking RNA polymerase from binding to the promoter as well, thus blocking the transcription of these genes. So when there's no lactose around, the Lac-I protein binds to this operator region, and by binding to this operator region, it is blocking the promoter from being bound by RNA polymerase. If RNA polymerase cannot bind to the promoter, we cannot get transcription. If we are not transcribing the Operon, we're not making proteins from it. And so this gene is repressed. The Operon is kept off. The Operon is repressed in the absence of lactose. It's repressed by the Lac-I Operon. But remember, this is a negative inducible system, so the natural tendency of the gene is to be on. When lactose enters the environment of the cell, those trickling amounts of permease that were sneaking through with that very, very, very low-level constitutive expression we just discussed, those small, small levels of permease in the cell membrane of bacteria let that lactose into the cell. Very little bit of it, but some of that lactose gets into the cell. Once in the cell, that very, very low-level amount of beta-galactosidase that's there because of that trickling level of expression from the Operon is there to take that lactose and convert some of it into allolactose on the way to trying to cleave it into galactose and glucose. But once made in these low levels, allolactose binds directly to Lac-I. And when allolactose binds to Lac-I, the Lac-I protein changes its shape. When it binds to allolactose, that makes the whole protein change its shape. And when the Lac-I protein changes its shape, it can't do what it used to do before. Lac-I was binding to the operator and blocking RNA polymerase because it had a particular shape that let it bind there. Once allolactose binds to Lac-I, it changes its shape, and that shape change causes Lac-I to no longer be able to bind DNA, and so Lac-I lets go of DNA and floats away. With the Lac-I repressor off of the DNA, the promoter is no longer blocked, and it is free to be bound by RNA polymerase, and we get Lac-Operon expression. Once the Lac-Operon is fully on, we express the Lac-Z, Lac-Y, and Lac-A proteins at ridiculous levels, so we make tons of permease, allowing tons of lactose into the cell. We make tons of beta-galactosidase, cutting all of that lactose into galactose and glucose, and the cell is effectively metabolizing lactose sugar. There's a movie that I've posted on the online resources portion of Canvas that shows all this. I know it's a lot to see in text. And again, I'm a pictures person. So if you really want to see this happening the way it happens in a normal cell, I invite you right now to pause this lecture, go log into Canvas, and watch that video and see how this works. The text also goes into some great detail regarding the effects of specific mutations in the lac operon. For example, what would be the effect of a mutation where LAC-I can no longer bind to DNA because of a mutation in the LAC-I gene? What would the effects of that be? Think about that. What would happen in the cell if LAC-I could no longer bind to DNA under any circumstances? What would be the effect of a mutation in LAC-I where its DNA binding ability was unaffected, but it could no longer bind to allolactose? What effect would that have on the regulation of the lac operon if this mutant LAC-I could no longer bind to allolactose? What would be the effect of a mutation to the operator sequence? So LAC-I was completely fine, but it still couldn't bind to the lac operon because its landing site, its regulatory element, that operator, had a mutation in its sequence. I want you to think about these questions. Think about these long and hard enough that you could comfortably answer them, that you could comfortably predict the effects of these mutations. Because I'll tell you right now, when we're done with this unit and done with this week, these are the types of questions that I want you very comfortable with. And these are the types of questions that you need to be comfortable with for our last unit exam. I know it's all new now. And I know you're probably very uncomfortable with these types of questions now, but make a little note for yourself. Bookmark this portion of the lecture. Be able to come back to this lecture at the end of the week, after we've done all of our activities and our review, and make sure you can answer these questions with confidence. These are the types of questions, and the answers to them, of course, the correct answers that you come up with, that are going to demonstrate to me, better than anything else, that you understand this material. So these are the types of questions that you can expect to see in the future. So that's lactose regulation. So just to recap, lactose regulation is pretty simple. When lactose is not around in the cell, the LAC-I repressor binds to the lac operon and keeps it off. No lactose to metabolize, no proteins for metabolism. When lactose is around in the cell, it's converted to allolactose. That allolactose binds to LAC-I. Now LAC-I lets go of the operon, and the operon is free to be expressed. Protein for metabolizing lactose is now made because lactose is in the cell. Sounds good. But remember the 2 plates of sugar we put in front of our bacteria? Remember we wanted to give bacteria a choice between glucose and lactose? Remember we said that when given that choice, bacteria will always choose glucose over lactose? So how does glucose figure into this regulation? Let's talk about that now. Everything we just talked about, to me, awesome, in the truest sense of the word. But we have this idea of choice. I told you that bacteria want the lac operon on when lactose is around. Okay, we've covered that. We see how that works. The lac operon can be on when lactose is around because we let go of LAC-I repression. But I told you that bacteria want the lac operon on when lactose is around and glucose isn't because they have that preference for glucose. And we haven't talked at all about a preference for glucose. So how do bacterial cells sense glucose availability? And how do bacterial cells regulate the lac operon based on glucose availability? The short answer to that question is a phenomenon called catabolite regulation. Catabolite regulation refers to any regulation at any gene in response to glucose levels. When glucose levels get low, cells get freaked out. That's bacterial cells, yeast cells, even your and my cells. Cells get freaked out when glucose levels are low because glucose is the best, easiest sugar to use, and if glucose levels are getting low, it means we might be starving. This is a life-threatening stimulus. If glucose levels are getting low, we are either going to die or we better get our cells metabolizing other sugars because if we don't do that, we're going to die. One molecule that's made in all stressed out cells is a molecule called cyclic AMP. It's a modified version of ATP. It's adenosine monophosphate, but it's a unique form of adenosine monophosphate that kind of cyclizes on itself. That's all chemistry stuff. It doesn't really matter. But this is a stress molecule for cells. When cells make cyclic AMP, that's like an all-points bulletin. That's a loud PA, public address announcement for the entire cell. Hey, we're in lots of trouble. And what happens when cells have high cyclic AMP levels, when they're all stressed out and freaking out, is they mobilize lots of energy stores. They gear up for a very stressful situation. And believe it or not, one of the main effects of caffeine on us is an artificial increase in cyclic AMP levels. What you're actually doing when you drink caffeine stimulants like coffee is you're fooling the cells into thinking that they're about to go into a very stressful environment. And to contend with that stressful environment, with those high cyclic AMP levels, they mobilize a lot of energy stores. And that's what tweaks us out. That's what wakes us up with caffeine. That's what tweaks us out and makes us hyper. So you drink caffeine, you fool cells into thinking that there's a stressor going on. The cells bump up their cyclic AMP levels in response to that caffeine and start mobilizing energy stores to deal with that stress that's completely artificial. The way this pertains to us is that low glucose levels are a stress for a cell. It's a life-threatening stimulus. It's a starvation stimulus. So when glucose levels are low, the cell increases cyclic AMP levels. As glucose levels drop, cells make more and more cyclic AMP. As that loudspeaker announcement, hey, bad things are happening, glucose levels are falling, get ready to deal with that stress. Cyclic AMP is a small molecule, and it binds to things. It binds to lots of different things in the cell, including many, many proteins. And one of the proteins that cyclic AMP binds to is called CAP. We will refer to CAP in that way from now on, but CAP stands for catabolite activator protein. When cyclic AMP binds to CAP, it turns CAP on. And when CAP is on, it binds to DNA. And the DNA it binds to is in the Lac Operon promoter. But it doesn't bind there to block RNA polymerase the way that LAC-I did. Instead, CAP binds to the promoter to help RNA polymerase bind to the promoter and to help initiate transcription. What I haven't told you so far about the Lac Operon, because I didn't want to confuse things, is that the Lac promoter is actually pretty crappy. And by crappy, I mean that it's not a very good landing site for RNA polymerase. For RNA polymerase to effectively and efficiently bind to the Lac promoter, it needs help. It needs to be forced to bind to that crappy promoter, and that help comes in the form of this activated CAP protein. So CAP, only when bound by cyclic AMP, is the protein that forces RNA polymerase to bind to the Lac promoter for transcription. So when glucose levels are low, cyclic AMP levels are high. When cyclic AMP levels are high, they bind to CAP. When cyclic AMP is bound to CAP, together CAP and cyclic AMP bind to the Lac Operon. When CAP binds to the Lac Operon, it recruits and helps RNA polymerase bind. When RNA polymerase binds to the Lac promoter, you get transcription and the Lac Operon is on. So to say it simply, the Lac Operon can be on when glucose levels are low. When glucose levels are high, the cells are not stressed out. They don't make a lot of cyclic AMP, and so there's no cyclic AMP to bind to CAP. When cyclic AMP is not binding to CAP, CAP is unable to bind to the Lac Operon. If CAP is not bound at the Lac Operon, there's no one there to help RNA polymerase bind. And if CAP is not there to help RNA polymerase bind, we do not transcribe the Lac Operon, and the Lac Operon is off. So in text, as glucose levels climb, cyclic AMP levels drop. The relationship between glucose levels and cyclic AMP levels are always opposite. As glucose levels go up, cyclic AMP levels go down. As cyclic AMP levels fall, CAP can no longer bind to the Lac Operon. Without that help of CAP, RNA polymerase no longer binds to the Lac Operon. The expression of the operon ceases, even if lactose is still available. And that makes sense, Because if lactose is available to the cell but glucose levels are climbing, the bacteria want to make that choice and use that available glucose. Bacteria will always prefer to use glucose if and when it's available. So here's a little table to summarize all of this. And while I rarely provide tables or figures in the notes, in the text notes that I provide on Canvas, because I do want you going to the textbook, I do want you taking notes on your own. That's also part of the learning process. The regulation of the lac operon is so complicated that I've given you this table in your notes so that you don't have to copy it. You can just kind of make notations as you need to and try to lock this material down in your mind. I'm going to go through this table with some considerable detail and some considerable time right now and try to break down for you each of these examples as they go. The layout of this table is almost like a Punnett square in the sense that where the columns and the rows meet is the condition we're talking about. In other words, right here in this area of the table, we're talking about here is when glucose is around and lactose is around. This area of the table is referring to when glucose is around and lactose is not around. This is when there's no glucose around and lactose is present, and this is when there's neither of both sugars. So first let's talk about when glucose is present and lactose is present. And let's think about this common sense first before we go into the molecular stuff. If glucose is present and lactose is present, in other words, we're putting two plates in front of bacteria, glucose on one plate and lactose is another, we're saying that the cells should prefer glucose. And in other words, the lac operon should be kept off. Because the cells prefer glucose, they don't want to express the lac operon. So now let's see what actually happens in the cell to make that possible. Well, if lactose is around, that means the cells can make allolactose, because lactose is present in the cell. If lactose is around and allolactose is made, lac-I will not be binding to the operon, so conditions seem to be favorable for lac operon expression. However, glucose is around, and when glucose levels are high, cyclic AMP levels are low. And if cyclic AMP levels are low, CAP is unable to bind to the promoter of the lac operon, and without CAP's help, RNA polymerase cannot bind, and in the absence of RNA polymerase, we get no lac operon expression. So that's how the choice is made. Now, when glucose is around and lactose isn't, we know we don't want to express the lac operon. We've got both things against us. The preferred sugar is around, and the sugar that the lac operon allows us to metabolize is not around. So we definitely don't want the lac operon on under these conditions, because we have our preferred food, and there's no glucose to digest anyway. But let's see what's going on at the molecular level. When there is no lactose around, the cell is unable to make allolactose. In the absence of allolactose, lac-I is free to continue binding, and so we get repression of the lac operon. Glucose levels are high. If glucose levels are high, cyclic AMP levels are low. With low cyclic AMP levels, we get no CAP binding, and without CAP binding, we do not help RNA polymerase bind. So we have the presence of repression and the absence of help for RNA polymerase, so we have two strikes against us and no expression. How about in the absence of both sugars? In the absence of both sugars, we have no lactose around. No lactose around means we don't make allolactose. Without allolactose, lac-I is free to continue binding. So we get repression from allolactose. With low glucose levels means cyclic AMP levels will be high. Excuse me. With high cyclic AMP levels, CAP will be able to bind. So CAP binds to the operator or the promoter of the lac operon, and it attempts to help RNA polymerase to bind as well. However, every time RNA polymerase tries to bind, lac-I is there and in its way. And so repression trumps activation, and the operon is kept off due to lac-I binding. So in all three of these scenarios, for different reasons, but in all three of these scenarios, lac operon expression is repressed. But here now, when there's no glucose around and lactose is around, The presence of lactose means that allolactose can be made. When allolactose is made, allolactose binds to Lac-I and stops Lac-I from continuing to bind to DNA. So repression is alleviated in the absence of Lac-I binding. But glucose levels are low. When glucose levels are low, cyclic AMP levels are high. Cyclic AMP complexes with CAP, allowing CAP to bind to the Lac-Operon, and when CAP binds, RNA polymerase is helped. CAP is there helping RNA polymerase to bind, and RNA polymerase is free to bind with that help because Lac-I is not there to stop it. Once RNA polymerase binds to that Lac-Operon, transcription proceeds and the operon is expressed. The operon is expressed only when lactose is around and glucose isn't. And those are the exact conditions by which we would want the operon on. A beautiful, beautiful, elegant system to allow bacteria, a simple cell as simple as bacteria, to allow that cell a choice of sugar to metabolize. We call this a gene circuit because we're dealing with multiple inputs from multiple pathways and coming up with a cohesive end result. I'm going to use the trip operon as a separate example for how operons can be regulated. The way that the trip operon is regulated is completely different than the lac operon, but no less respect-worthy. When we're talking about sugars, such as lactose and glucose, we're talking about food. And we want the genes needed to eat these foods or metabolize these foods when those foods are around. In other words, there's no point having the lac operon on when lactose is not present to eat. We need the operons on to metabolize those sugars. Why try to eat a sugar and metabolize a sugar that isn't there to be eaten? But the opposite logic applies when we're talking about biosynthesis. When we're making biomolecules, such as amino acids, such as tryptophan, we want the genes that are needed to make those biomolecules on when the biomolecules themselves are not around. In other words, if we need tryptophan and we have to make tryptophan, why would we make tryptophan when tryptophan levels are already high? We only want to make tryptophan when tryptophan levels are low. It's the opposite logic as the sugar. So always keep in the back of your mind what the molecule you're dealing with is and what the cell wants to do with it. If the molecule is a sugar, the cell wants to metabolize it. If the molecule is an amino acid, the cell needs it on hand and either will consume it or make it. So that's a fundamental difference behind the regulation of these operons. And if you can understand that difference, you can kind of sort out the regulation a whole lot easier. I'll often ask on quizzes and exams with these types of questions, does this make sense? And when I mean does it make sense, what I'm getting at is, does it make sense that the cell expresses the trip operon when tryptophan levels are low? The answer is yes. The trip operon allows the cell to make tryptophan. And the cell only wants to make tryptophan when tryptophan levels are low. If tryptophan levels were high, why in the world would the cell want to make more? It already has enough on hand. So I've already said it, but the trip operon is a different operon, distinct and separate from the lac operon, and the trip operon contains genes encoding proteins that are needed for the biosynthesis of the amino acid tryptophan. And the cell only wants this operon on, and it only wants to express these genes when tryptophan is not around in the cell. So the trip operon is an example of a negative repressible system. The natural default state of the operon is to be on. It must be forcibly turned off, but we're saying it's repressible now. So most of the time the cell allows the operon to be on, and it only turns it off under distinct and unique circumstances. So how is this achieved? How is the trip operon regulated so that it is only expressed when tryptophan levels are low and only repressed when tryptophan levels are high? And the answer to that in one word is attenuation. Attenuation is a regulatory mechanism that relies upon the premature termination of transcription at the trip operon to be functional. At the very beginning of the trip operon, before any of the structural genes for the proteins involved in trip biosynthesis, at the very beginning of the trip operon is a sequence of DNA called the leader region. And the trip operon, when transcribed, of course the first part of the operon is what's transcribed first, so the leader region is what is first converted into a messenger RNA. So this is the basic layout of the leader region of the trip operon. You see the trip operon itself, all of the structural genes of the trip operon are off screen here. We're only focusing in on the leader region. And the leader region is made up of 4 distinct regions, 1, 2, 3, and 4. The leader region does not encode a full protein. It does not have any functional significance in translation. It is simply the regulatory region of the trip operon. The way that this leader region is sequenced at the DNA level and at the RNA level when we transcribe it is that it can form 2 different RNA base paired structures. Call these stem loop structures. I'll show them in just a second. One of these structures promotes the premature termination of transcription of this operon, and the other allows transcription to continue and does not promote premature termination. Again, we have 4 different regions or sequences of interest in this leader region. If left completely alone, the very first sequence of the leader region to be transcribed is going to be region 1. The very next sequence is going to be region 2. And region 1 and region 2, when they're part of single-stranded messenger RNA, can base pair together. In other words, where there's an A here on region 1, there's a U here in region 2. Where there's a C here in region 1, there's a G here in region 2. If there's a U here, there's an A here, and so on. So they base pair. The next region to be transcribed would be region 3, and the region after that to be transcribed would be region 4. And region 3 and region 4 can also base pair with one another the same way. And this 1 base pairing with 2 and 3 base pairing with 4 is responsible for premature termination. If this structure is allowed to assemble, transcription stops at that point, RNA polymerase falls off of the operon, and remember now, the structural genes, the genes that are important for trip biosynthesis, are off screen. We haven't even come close to transcribing those yet. So if we prematurely terminate transcription, if we fall off of the operon at this point, based on this structure forming, we never transcribe the rest of the operon, and we never translate those genes. So we call this 1 base pairing with 2, 3 base pairing with 4, stem loop structure, base paired structure, a terminator, because it terminates transcription. However, there's another structure that can form. If somehow you could pry region 2 away from region 1, and if you could somehow pry region 4 away from region 3, region 2 and region 3 can also base pair with one another. And when they do, this structure does not promote premature termination. And so transcription by RNA polymerase will continue down the entire operon, and the entire operon will be expressed. But remember now that we need to transcribe all these regions in one shot. So as transcription of the operon begins, first region 1 is going to be made, and then region 2. And as soon as region 1 and region 2 are made, their tendency will be to base pair with one another. So how do we ever make the region 2, region 3 base pair structure if region 1 and region 2 are transcribed first? I'd like to remind you that there are no nuclei in prokaryotes. One of the main defining features of prokaryotes is that they do not contain a nucleus All components of the cell float equally in the cytoplasm. And so transcription and translation can occur simultaneously in prokaryotic cells, because as soon as the messenger RNA is transcribed, ribosomes are around to latch onto it and begin translating. In our own cells, transcription only occurs in the nucleus and translation only occurs in the cytoplasm. So both transcription and translation, they cannot occur at the same time in our cells because the nucleus provides a boundary. But there's no nucleus in prokaryotic cells, so there's nothing stopping transcription and translation from occurring simultaneously. In this leader region that we've been discussing, in part of region 1, there is a start codon to initiate translation, and there are lots and lots of trip codons calling for the amino acid tryptophan. If tryptophan levels are low, however, there's not going to be much tryptophan amino acids present in the cell to build on that growing protein chain. If tryptophan levels are low, they're low. And even though codons are calling for tryptophan, there's not going to be much tryptophan around to add to that growing chain of amino acids. Because of this depletion of tryptophan, the ribosome is going to stall at that point in region 1, pausing and waiting for more tryptophan to come and enter it. With the ribosome stalled there in region 1, that ties region 1 up and makes it unavailable for base pairing with region 2. Oh, I love it. Since region 1 is tied up, region 2 remains unbase paired. It remains unbase paired until region 3 is transcribed. Once region 3 is transcribed, region 2 and region 3 pair up. That is the anti-terminator form that allows transcription to continue. RNA polymerase continues on, transcribes the rest of the operon, so that those proteins of the trip operon can be made to make more tryptophan because tryptophan levels were low. If tryptophan levels are high, what's going to happen? Well, if tryptophan levels are high, region 1 is going to come out of the RNA polymerase. The ribosome is going to latch onto it, but because tryptophan levels are high, the ribosome is going to blow right through that region 1. It's going to encounter the start codon, it's going to add tryptophan, added onto the chain because those levels are high, and then the ribosome is going to fall off of the leader region because there's a stop codon. Since the ribosome is able to keep up with the RNA polymerase, there's no pausing, there's no stalling, there's no stopping of the ribosome on region 1. Region 1 is free to pair with region 2. When region 3 emerges, region 2 is not available for base pairing, so region 3 remains until region 4 is transcribed and the region 3-4 base pairing structure forms. This is the terminator. When region 1 base pairs with region 2 and region 3 base pairs with region 4, we create the terminator structure, which stops transcription cold in the leader region, never allowing RNA polymerase to transcribe the trip operon genes. Why? Because tryptophan levels were high, and we don't need those trip operon genes transcribed. This is the image from your textbook. When tryptophan levels are low, RNA polymerase chugs down the leader region, and it doesn't stop. It continues down the leader region. Once the region 1 is transcribed, a ribosome latches onto it and begins translating the short protein sequence called for there. That protein sequence, however, calls for many, many tryptophans, and if tryptophan levels are low, the ribosome slows down and stalls at those trip codons because there isn't much tryptophan in the cell to put tryptophan onto this growing chain of amino acids. With the ribosome stalled on region 1, region 2 has no one to base pair with. As the RNA polymerase transcribes region 3, region 2 base pairs with region 3. This is the anti-terminator structure. Nothing stops RNA polymerase from continuing transcription, and it continues on down the operon to the structural genes that are responsible for making proteins needed for trip biosynthesis. So the cell will eventually make more tryptophan because the entire operon was transcribed when tryptophan levels were low. When tryptophan levels are high, the story starts the same way. RNA polymerase begins transcribing the leader region. Region 1 emerges first. The ribosome latches on to leader region 1 and begins translating it based on the sequence it calls for. It translates that sequence, it encounters the trip codons, but this time the trip codons do not cause any pausing or stalling because there are abundant levels of tryptophan in the cell. The ribosome continues on, either translating the leader region or it falls off. It's really immaterial, because what is important is that as region 2 emerges, region 1 is free for base pairing. As region 3 emerges, region 2 is already complexed with region 1, so region 3 has no one to base pair with. Once region 4 is transcribed, region 3 base pairs with region 4, and this is the termination structure. The region 3-4 base pairing causes RNA polymerase to immediately fall off of the trip operon and not continue transcription. Transcription is not continued. The trip operon is not expressed. The proteins for trip biosynthesis are never made as the cell wishes because tryptophan levels are already high. Why would you make more tryptophan when you already have plenty on hand? This provides an extra layer of regulation, allowing bacteria to change its mind and abort gene expression even though transcription has already begun, because the cell has realized that tryptophan levels are high. What a beautiful system. What a beautiful system. So what did we talk about today? We covered quite a bit, didn't we? Some fairly complex topics now, bringing us into this final leg of the course. We started off talking about some fairly simple conceptual ideas of regulation. Positive control, where the natural tendency of a gene is to be off and something has to be done to turn that gene on. We call that an activation event of a gene. We used a car as an analogy. And negative control, where the natural tendency of the gene is to be off and something needs to be done to... the natural tendency of the gene is to be on and something must be done to turn that gene off. The gene must be repressed through repression, and we used a hot water heater as our example for that. We went on to define operons. We said bacteria cluster their genes of similar function for a similar cellular process together into operons. It makes regulation easier. It allows the cell to coordinate its regulation. Operons are under the control of a single promoter, and the different genes of an operon are transcribed as a single messenger RNA. We then went on to define these terms of not only positive and negative regulation, but repressible and inducible regulation, and we saw that we have operons that come in all four types: positive repressible, positive inducible, negative repressible, and negative inducible. And I do want you to be comfortable with all four types and know how they behave, how they're regulated, and what they mean. And we then went on to our example of sugar metabolism. We made the point that all living cells on this planet prefer glucose over any other fuel or food source, and that means that those cells need to be able to make a choice. When given two different carbon sources, two different sugars, glucose and another, cells should always choose glucose. Bacteria are no exception. If you offer bacterial cells glucose and lactose, bacteria will always choose glucose. We said that 3 genes make up the lac operon: the lac-Z gene, the lac-Y gene, and the lac-A gene. We defined what these genes were. Lac-Z encodes beta-galactosidase, which cuts lactose in half. Lac-Y encodes a permease, which brings lactose into the cell. There's a 4th gene that is not part of the gene cluster. It's the regulatory gene for the lac operon. It encodes the protein lac-I. Lac-I is a repressor. We went over the lactose regulation of the lac operon. In the lactose regulation of the lac operon, the presence of lactose triggers the formation of allolactose. When allolactose is present in the cell, it binds directly to lac-I. Lac-I bound to allolactose cannot bind to the lac operon, and so repression is alleviated in the presence of lactose. If there's no lactose in the cell, we don't make allolactose. In the absence of allolactose, Lac-I is free to bind to the Lac-Operon, and Lac-I is a repressor, so when bound to the Lac-Operon, Lac-I turns that operon off. That was the lactose regulation story. But we wanted this choice. We wanted glucose involved. And so we had to talk about catabolite regulation, which is the controlling of gene expression through glucose availability. We made the point that when glucose levels are low, this is a stress to the cell, so cyclic AMP levels climb on that trigger. We see high levels of cyclic AMP when glucose levels are low. Cyclic AMP binds to this CAP protein, catabolite activator protein. When cyclic AMP binds to CAP, it turns CAP on. And CAP, when turned on, binds to the Lac-Operon promoter and assists RNA polymerase binding. And so we covered the glucose regulation of the Lac-Operon. If glucose levels were high, cyclic AMP levels were low. Without cyclic AMP levels, there was no CAP binding. With no CAP binding, RNA polymerase could not be helped, and we did not see transcription. But if glucose levels were low, cyclic AMP levels were high. With high levels of cyclic AMP, CAP could bind, and if CAP was binding, then RNA polymerase binding could be helped, and the Lac-Operon potentially could be turned on. We then synthesized these two different regulations and talked about how the interplay of glucose and lactose availability led to the ultimate regulation of the Lac-Operon. We closed the lecture with a discussion of the trip operon and specifically attenuation, this idea of region 1 and region 2, region 3 and region 4, serving as a terminator sequence for premature termination of transcription, repressing this operon. But the whole idea of coupled transcription and translation, if we can get that ribosome to stall on region 1 due to low tryptophan levels, then we tie that region 1 up, forcing region 2 to base pair with region 3. That's the anti-termination structure that allows transcription to continue, and we express the entire trip operon in the presence of low tryptophan levels, exactly as we want them. So that's all we're going to talk about for gene regulation in prokaryotes. We're going to move ahead now to the quantitative genetics portion of the course in the next lecture. Earlier in the lecture, many of you were asking me, well, what about eye color in humans? What about height in humans? Why do humans look blended when pea plants look like one parent or the other? How do you bring Mendelian genetics into our own blended appearances? And that is what quantitative genetics is. So we'll start to see how Mendel's rules hold up, but still create this spectrum of traits that we see in complex organisms such as our cells. But that'll have to wait for the next lecture. As always, thanks for watching.